Thanks everybody for coming out. As you can see by this turnout and the diversity of partners up here on that board, this is a hot topic and something that really concerns all of us. This event was spearheaded tonight by the BCWF and the East Kootenay Wildlife Association, Glenn's group. The other major partners are ourselves, Backcountry Hunters and Anglers, the Big Game Records Club of BC, the Kootenay Wildlife Heritage Fund, and then some smaller donors from actually all around the province. We have the Karamias Costin Sportsman Association, Lumbian District Wildlife Association, the Osceola Fish and Game Club, and the Summerland Sportsmen's Association. So really thankful for all those partners that brought this together for us. I'm going to be the moderator tonight. I'll introduce the speakers and I'll try to run the Q&A. There'll be Q&A after each speaker and then at the end for a general Q&A. So just put your hand up when that time comes and we'll try to answer your questions. First speaker tonight is Jessica Russell. She's a response planning specialist. Jess has spent the majority of her career managing and planning responses for large disease and invasive pest incursions at the national level in New Zealand, and now she's here in Canada. She's experienced in translating disease management theory into application and planning responses to primarily animal health threats, but also plant pests, foodborne outbreaks, and adverse events. These include leading the eradication of Gumboro disease from poultry in New Zealand, planning and overseeing the response to New Zealand's largest outbreak of Salmonella, and developing the management research program for the newly introduced granulate ambrosia beetle. Within Canada, she has supported the planning epidemiology unit of the Western High Path AI response with the Canada Food Inspection Agency and supported the incursion planning to CWD here in BC along with the provincial team. So warm welcome for Jess, please.
You're just going to have to watch me fiddle with this for a second. All right. Is that working? Perfect. Hi, everybody. Can everyone hear me just to start? Awesome. Okay, be a bit louder. Fabulous. So hi, thank you so much for having me. This turnout is amazing. There's a lot of people in this room. There are so many people standing and still coming in. So really impressed at how interested everyone is here in chronic wasting disease. So the BC Wildlife Federation has asked me as an external independent person to come in and talk to you today about chronic wasting disease and look at it from an objective third-party perspective around how we can plan for this kind of response. So I am a response planning specialist, so as Ellen very kindly introduced me, I look at taking technical information and applying that in a more practical approach. So we tend to use emergency planning principles or military planning principles to effectively treat incursions of diseases or pests similar to how you might treat an enemy force. So we need to take technical information around how the disease might act, its biology, and then use that to inform what we do in order to get ahead of it or get rid of it. What we find in a lot of these incursions is that if you're in a place where you've never had an incursion like a disease, it's never been in an environment similar to where you are, it makes it really hard to plan. You don't know how that disease is going to respond to the environment. It might be in a new host. You don't know what the biology is going to do, and it makes it really difficult to sort of figure out how you're going to get ahead. Lucky for BC, BC is a bit of a late adopter of chronic wasting disease, you could say, and so there's a lot of other examples of where chronic wasting disease has been for a long time, and we can take that information and learn from it. So what I'm going to be doing is going through some other jurisdictions, looking at what they did for their response, what worked and what didn't, and then what that means for the planning of the BC response. I am from New Zealand, so I have a weird accent. So if you don't understand anything that I say, please feel free to ask clarifying questions at the end, and I'd be more than happy to repeat the information that was not clear. Awesome. So Firstly, I'd just like to also acknowledge that we are all gathered here today in this room on the unceded lands of the Katunaha First Nation, who have looked after this land for a really long time. So really grateful to be here today with all of you, but also on these beautiful lands. So, our agenda for the day. So I'm going to take you over a brief overview of chronic wasting disease biology. Given that you're all here because you're interested in CWD, you probably know what it is. I'm not going to do too much detail of this, but I am going to highlight some of the key principles that we need to remember and acknowledge as we go through our planning. I'm then going to talk about disease management principles as a whole. So what kind of things do we need to focus on? What are important to keep in context as we move forward? And then I'm going to go through examples along the scale of action. So looking at all of the different jurisdictions, there are a number of them, so we're not going to do all of them, but I have selected a few that I think provide some good lessons. Figure out what worked and what didn't, and then talk about how that has built a best practice portfolio for CWD across the world. Lastly, we're going to talk about what does uncontrolled chronic wasting disease actually mean? A lot of people kind of say, oh, it's going to be bad, but we're going to go into what that actually looks like. And then how do we apply this to BC? Is it too late? Can we get ahead of the spread? What does it mean? So we'll get into it. So chronic wasting disease is a fatal prion disease that affects cervids. As you'll know, it is a misfolding of the prion protein. So up on the right here, we've got a normal prion protein, and then we have a diseased, misformed, twisted protein. So the way that chronic wasting disease works, like other prion diseases, is that one prion will spontaneously, or triggered by another one, mutate itself. It bumps into another protein and it mutates that protein. You end up with a daisy chain effect where you end up with clusters of misfolded proteins. Interestingly, prions are found along nerve axons and at presynaptic terminals. So what does that mean? It means on nerves. Where do you have a lot of nerves? In your brain and in your spine, and so therefore we end up with accumulation of prions that have folded and twisted along those areas. So we end up, when you look at it under a microscope, this is healthy nerve tissue. Oh, I've been told not to stand in front of the projector, sorry. Got healthy brain tissue and then infected brain tissue. So you end up with these holes that causes a number of symptoms in infected cervids, including weird behavior, losing weight very quickly, hence the common name of the wasting disease. So this is called a transmissible spongiform encephalopathy. It's a very long phrase. It's in a disease family similar to mad cow. There are a number of these species, or Creutzfeldt-Jakob disease in humans. With CWD, one of the big issues is that incubation takes a really long time. So incubation is the time that a disease is in the body but not showing symptomatic clinical signs. With CWD, this can last anywhere between 16 and 36 months, meaning that we have silent carriers, where a diseased cervid can look completely healthy. During this time, they can shed infectious proteins or prions into the environment and create reservoirs of disease. So one of the issues as well is that the time from oral infection to death, so that is when they come in contact with prion proteins up until the time they die, is anywhere between 23 months in mule deer, and it's a little bit longer in elk. So it sounds like a long time, but it can act really quickly once those symptoms appear. So once they appear, normally it's about 3 to 4 months, particularly in whitetails. But there was an example relatively recently where a 6.5-year white-tailed doe, she weighed 165 pounds in Wisconsin. She was captured, had a collar put on, and then 1 month later she was recaptured, found to be positive, and she weighed the same. About a month later, I think, what do my notes say? 8 weeks later, she was recaptured, and she was dead, and she weighed 80 pounds. So she halved her weight in 8 weeks after the disease symptoms progressed. So it's really fast sometimes. So while that time of incubation is really long, the actual disease can kill individuals relatively quickly. So there's been a number of studies done around what this looks like on the population or herd level, and what it's summarised as is infected CWD deer are 4.5 times more likely to die per year than a non-infected individual. So that's really big. Down on the bottom here, we've got a annual survival probability chart from Minnesota, and they did a study looking at the different life expectancy for infected and uninfected males and females. This is in mule deer. And they found that in general, a female would, when uninfected, they had an 83% likelihood of surviving to the next year. So that's pretty standard. They could otherwise have died from predation, other injuries, other diseases, just natural causes. When they were CWD infected, that was halved to 41%. And in males, it's a lot worse. So they found that normally males had a 69% chance of survival, and that went down to 17% when they were infected with CWD. And that's just year to year. So we're going to have some big impacts, right? So the disease is transmitted through prions. As mentioned, these are those proteins that have twisted, and they are practically indestructible. Makes it really difficult to manage. They can remain infectious for more than 10 years. Bleach doesn't get rid of them, cooking doesn't get rid of them, burning doesn't get rid of them. So it makes it really difficult. Once they're in the environment, they are there for a long time. It's looked like at the moment that it's more than 10 years and they're still viable to reinfect. So once we've got them in the environment, it's really hard to get rid of them. So one of the key things I want to talk about is the way that the disease transmits from one animal to another. So in early incursion, the disease tends to be direct animal-to-animal contact. So that is through grooming, marking, mating, because prions are found in bodily fluids. So if a deer comes in contact with another infected deer, it's going to spread those prions to that other animal. That's what we see in an early incursion when we have low levels of prions in the environment. However, once established, we get more of this indirect infection. So what that looks like is on the right-hand side, we have more carcass decomposition that are infected with prions in the soil, more animals touching that soil. They then excrete prions through their urine or their feces or their blood, and then more animals come into contact with that again. So it ends up being like a little negative feedback loop. What I like to think about is like a bathtub spiral or like a vortex. So up on this right-hand side, you can see how when you start, it's really wide. So there's a lot of distance to cover the vicinity of the spiral, and then it gets tighter and tighter and tighter towards the bottom. So in early incursion, that spiral is slow, really slow as it's getting increasing with more and more infected animals, and then it starts getting quicker as we get a more intensive load of prions in the environment. So it means that once we're in later incursion, that transmission really speeds up. Oh, I feel like this might be working again. Okay, fabulous. So males are more likely to be infected up to 1.5 to 3 times more likely than females. There's also been more, there's been recent studies as well to show that there's more evidence towards vertical transmission. So that is, thank you, does passing CWD onto their fawns in utero. So that's not good. And we've seen evidence that the population starts to decline once there is 13% prevalence in the environment. So that is, for all of the samples that you take, 13% were positive. And Holger, I believe, is going to go through how that is calculated. Oh, got some feedback. Awesome. So speaking of transmission, do we know what where that's coming from? Oh, no, it stopped. Okay, I think we're good. So this map that the USGS produces is really useful. They update this every month. I highly recommend looking at it if you are interested. So the dark grey is where So the grey squares is where CWD is in wild cervids. Chronic wasting disease is now found in 36 out of 50 states in the US, so that's a lot. Recently, Washington was found to be positive in 2024, and Indiana in 2024 as well. And recently in January, Georgia state also got their first detection. Internationally, close to home, it's found in 5 provinces in Canada, including Quebec, which has it in its captive populations. And it's also found in Finland, Norway, Sweden, and in captive populations in South Korea as well. So that distribution is expanding more and more and more. So what are the tools that we've got in order to detect CWD? So gold standard is post-mortem testing, where we are able to take lymphatic tissue from the lymph nodes or from the brain stem, and we're able to then do an antigen detection via an ELISA screening, which is a laboratory process where they can test whether that exists in the tissue or not. And then in order to confirm that, we look at those little pink bits of tissue and we see whether there's holes in it or not. And that is used as considered to be the gold standard, and it has to be done at an accredited laboratory. There are some evolving techniques using antemortem testing, so that is while the animal is still alive as a surveillance mechanism, using either rectal or tonsil biopsies. The problem is that if you manage to do a biopsy and you get the wrong piece of tissue by accident, you might not know, the prions just might not be there yet, you might end up with a false negative, which means that you make decisions when you should have made different decisions because it was actually positive. Moreover, there are some more novel methodologies coming out, including something called RT-Quick, which is very, very sensitive. It can detect it in soil and it can detect it in other environmental samples. So that is something that is moving forward really quickly, but it's not yet approved for actual decision-making testing at this stage. Additionally, dogs are being trained to smell out prions, because dogs are amazing, so why not utilize them? And that's in the process as well as becoming more reliable. There are no known treatments for any TSE disease. That includes the human disease. So some people say, oh, there's no vaccine, it's because no one cares. People do care because it's in people, right? We have Creutzfeldt-Jakob disease that affects a number of people every year. But because prions are proteins, they don't have unique genetic material that we can utilize to make vaccines, because normally a gene or some other kind of bacteria or something will have these kind of information that we can make a vaccine with. There are a lot of different groups working on vaccines yet, so it could be coming soon. We just have to keep our fingers crossed. There are some resistant alleles and different gene variations that provide some level of resistance to chronic wasting disease. However, it's not immunity. It's just that those deer might last longer before they die, or it might reduce how quickly they get infected with chronic wasting disease when exposed. Now, I've got a couple of key sort of articles up here around the risk, given that there is no vaccine of any kind for chronic wasting disease. I'm not going to go into the details around the risk for humans, but it has been known that prions are still detected in cooked, raw, or processed meat from CWD-infected animals. We don't know whether that is going to at some point jump to humans, but can I just say you don't want to be the first? So would highly recommend that you don't eat meat that has not been tested for chronic wasting disease from an area with chronic wasting disease. So, disease management principles in general. So, disease management, we look at effectively what is feasible, what are the limitations in the situation that you're in, and then look at some of that textual or contextual info to compare against, to figure out what you can do. And we utilize tools such as the Planning P. This is from the Coordinated Incident Management System that we use in New Zealand. There's also a similar one for the Canadian equivalent coordinated system. Effectively, it's a cycle. So you have the initial incursion down the bottom stem of the P. You figure out some objectives that you're trying to achieve, you develop your plan, you execute the plan, and then you have to stop and work out whether that plan actually worked. You're not going to do it once, you're going to keep planning based on the change of situation that you get. We also need to look at how are you going to achieve your objectives, and as new tools come up, you change your plan knowing that you have new tools. In general, when we've got a biosecurity incursion, it does tend to follow what we like to call the invasion curve. So along the bottom, we've got time. Along the top, we've got costs or the area infested. We tend to look at prevention as being a key component. And over time, the options that we have tend to decrease. So when we have an initial incursion, the opportunity to eradicate potentially exists depending on the biology of the organism. After a certain point, that's no longer feasible, and then you're trying to contain. From there, if containment is not possible, you kind of have to work out how you're going to live with it just expanding and enjoying itself in the environment. So therefore, one of the key things we want to do is always maintain options. You don't want to make a decision that is going to close off one pathway. Let's say you've got a little tree of options. You might go down here, here, or here, noting that if you go down the right-hand side, you might completely cut off your ability to ever go to the left. So therefore, maybe it's a good idea to stick more towards the middle, just in case new options come up in the future. So there are always going to be trade-offs as well in decisions. So one of the issues we get sometimes is, what about this one individual animal? What about them? It's a good point. What about them? And so we have to look at what are our priorities, what are those objectives? Are we looking to save the individual or the population? Are we looking to be willing to put up with short-term impacts, or are we looking for no impact at all and a less of an outcome? So there are some trade-offs we have to think about when we do this kind of planning. Okay, so the toolkit. Can everyone still hear me, just by the way? Fabulous. Okay, awesome. So what is in the toolkit for utilising management for chronic wasting disease? So just for one, we know already that reducing direct transmission is really important. So that is reducing density of animals in CWD positive areas in a non-selective way. We just want to overall reduce how many animals are touching each other, how many animals are pooping and other animals are touching it. We also then want to think of, we could be more selective. So removal of infected animals or those most likely to be infected. So for example, we know that male animals are more likely to be infected than females. So maybe we look towards a selective approach. We also want to prevent human-mediated spread. So because we know that animals who congregate together tend to get more infected, we want to reduce artificial congregation. So things such as baiting, luring, feeding. And then also preventing introduction into new areas. And this includes moving carcasses or perhaps fencing. And then our third tool is surveillance. And one of the great things about chronic wasting disease is that when we remove animals from the environment to achieve number one, that works as our surveillance. So we can do a double whammy where we get rid of potential vectors, but we also can see how well we're doing, how big is that prevalence. So what this kind of tends to look like on this very pretty rainbow arrow is that we've got a number of different management scenarios moving from lower intensity all the way up to higher intensity. So on the green side, we have monitoring only, so that's just doing surveillance, but we're not even really going to mandate anyone to submit samples. We then have management where we have mandatory submission, and that's it. So we require in certain areas for samples to be submitted, but we don't do anything else. Then we move forward into, we do a hunting-based approach with mandatory submission and limited sharpshooting or limited targeted removal on top. Then we go into hunting with regular targeted removal all the way up until wide-scale culls for an eradication approach. And so I'm going to take you through a number of these sort of examples, and there's a lot to cover, so I'll try and go quick, but that gives us our sort of summary of chronic wasting disease as a disease that we're trying to battle. So, Colorado. So This was the first ever detection of chronic wasting disease and is considered the home of chronic wasting disease. And it was first detected in captive deer in 1967, and then in 1981 it was found in the wild at approximately 0.29% prevalence. No one really cared about chronic wasting disease, to be honest, until the mad cow outbreak in the UK in 1990s, where people realised that actually chronic wasting disease is also a prion, and oh my god, what if that affects people? So the US federal government provided a large increase in funding at this time, and that meant that there was more interest and more samples voluntarily donated. So up in the right-hand side, we have a map of where chronic wasting disease is in Colorado. Everything that is not white is where chronic wasting disease is, so it's really spread to all of, pretty much all of the counties in Colorado. The darker the colour, the more prevalent they are, and it's around about 25%, I believe, up in that top right corner. So the Table Mesa herd was found to have reduced 45%. Over a 20-year period, where there was a really high density area of about 41% prevalence. Unfortunately, while there was still a lot of funding, so the state had a lot of money to be able to process samples, utilise labs, send out messaging, hunters didn't really know what they were doing. They didn't really see the value in it. So there was a really reduced amount of samples submitted between 2003 and 2017, which made it really difficult for Colorado state authorities to actually know what they were dealing with. So in this time, when they restarted sampling again in 2017 more seriously, some herds had increased prevalence by 10 times, which is really not ideal. Now they have released a rotating plan to do mandated submissions. So different areas across the counties will be submitting on a mandatory rotating basis so they can understand more about what they're dealing with. This is a really good on the bottom side, the squiggly line. So this is a summary of all of the herds in Colorado that are positive, and they were plotted all on a graph. A number of scientists way back in 2006, I believe, figured out that they expected an exponential curve, which is that dotted dashed line that's going up, and they plotted all of those different levels of prevalence over 40 years. And they found that while it didn't follow an exact upward slope like that, it did start to climb. And so this is a really good example of what chronic wasting disease looks like. We have an overall trend, and it kind of looks like that global temperature graph that you get shown all the time, where it squiggles up and down, but overall is going up. So in Colorado, there was also no sharpshooting of any kind, no targeted culling, and they're only just starting to do mandated testing. I would say this wasn't overly successful, personally, but that's up to the eye of the beholder. Wyoming. So Wyoming is a really good example where there has been a lot of research interest. So chronic wasting disease was detected there only 4 years after its neighbour in Colorado, with an initial prevalence of 0.9%. Again, little funding interest up until 2000, and then they got more money. They did a little bit more. They really looked at how to get more samples in. This increase down the bottom, we can see total samples tested by year in Wyoming from 1982 all the way to 2019. And you can see there's not a lot, and then it spikes, and then people get bored again, and it drops back down. So in 2018, they've started a more formalised submission process where it looks at key priority areas, but it's not mandated. It's just, where do we want to look? So in terms of some key examples in Wyoming, so one white-tailed deer herd in Converse County saw a 50% reduction between 2001 and 2009. That's only 8 years to have 50% of that herd decline. That's really not great. And it was an annual reduction of around about 10% per year. A mule deer herd in a similar area, so quite close to Converse County, dropped 21% annually. but 4 years between 2010 and 2014 in an area with a chronic wasting disease prevalence of 30%. Now, the project mule deer herd, which is this dark blue bit in the top map, that's got looking up to 65% chronic wasting disease prevalence. So that is 65% of the samples that you look at end up being positive. So given that we know how quickly it can kill deer, again, not looking great, so it's not ideal. But what this does mean is there's a lot of research and a lot of data. So this is quite a busy graph, and hopefully you can see it on the screen. But each of those squares is a different herd. The little circles are the number of animals tested, specifically males tested. And the bigger the circle, the more animal males were tested. This is a mule deer as well. And the colour of those circles is about the proportion of males of that herd. So you might have 0.2, so that is 20% of males, up to the red, which is 50%. So what we can see is that in areas with small circles that are more green, such as the South Converse up on the top left, we end up with a higher prevalence, which is indicated on the y-axis on the left-hand side, sorry, the right-hand side. And the more animals were tested that represented the higher proportion of males of that area, the lower the prevalence was. So if we look at Black Hills, they often were looking at getting rid of between 35% and 50% of the bucks in that herd, and they kept prevalence really low. So in comparison, South Converse, they had fewer tested and they represented a smaller proportion of bucks in the herd, and prevalence stayed significantly higher. So it's a sad story for some of those herds, but it does give us some really interesting data to look at. Okay, closer to home, I'm going to talk about Saskatchewan. This is also not a great news story. So Canada imported CWD, unfortunately, from South Dakota in the 80s into a captive deer herd, and they unfortunately escaped at some point, or CWD got leaked out somehow to the wild mule deer at approximately 0.18% prevalence. This was in 2000. They have implemented to date transport restrictions only. There is no mandated testing in Saskatchewan. There are encouraged areas where they encourage samples to be submitted, but it's not mandated. So South Saskatchewan River Valley, so where you can see the river coming in right next to the Alberta border, that has some of the highest CWD prevalence in mule male deer ever in the world, sitting at 92%. So that means that if you hunt a male buck, mule deer, if you hunt a mule buck, there is a 92% chance that that animal has CWD. And when we think about the impacts, that means pretty soon most of those animals will be dead. It's really, really not good. What we're seeing already, and the government is reporting it in Saskatchewan, is that those populations are really declining in that South Saskatchewan River Valley area. There are also increased detections in moose and other species, and it's slowly creeping northward. So in this colourful map down the bottom, this is where CWD is detected in mule deer, but they're also finding that it is now considered endemic in that coloured area, but it's creeping up into the boreal forest in the north. So Saskatchewan is a really good example of what happens if you just kind of let it do its thing. It does increase and it kind of explodes in that really intensive extrapolating kind of way. And I will say, and I should have said it earlier, is that a lot of these examples are taken from publicly available information, and a lot of different governments provide different levels of information. So it's not consistent between what Saskatchewan Online produces and allows individuals to look at. They might show prevalence in one particular area only. Other states will show it across the entire state average. Some show only in mule deer, some show only in white-tailed deer, some show it in moose only or only males, which makes it really hard to do an apples to apples comparison, but we're trying today. So across the border from Saskatchewan, what do we have? We have Alberta. So it was detected in farmed elk in 2002 and then found in the wild deer population in 2005 at approximately 0.18%. So the Alberta government was proactive. They did worry about the risk of Saskatchewan-based CWD creeping across the border, and they actually conducted the only proactive culling to try and reduce deer density in that area. It was pretty focused on what they thought were the high-risk areas, and then they found it further south. Unfortunately, they targeted slightly the wrong area, and that's not something that's in their control. So what they did in between 2005 and 2009 is they did what they called a 1-2 punch approach. So they did hunter surveillance, and then they did a winter response where they went in and culled approximately 2,000 animals per year after the hunting season. So what does that look like? Well, on this overall prevalence graph, you can see that between 1998, where they were doing those samplings, there was 0, and it very slowly started to creep up in that positive, and they kept it really low. They kept it below 0.2%. So if you can't see, it slits along the bottom of that graph, and then it jumps up. And what was the change? They stopped that culling program. That culling program was really unpopular. There wasn't a lot of collaboration with hunters. There wasn't a lot of communication with the public either. And so they were forced to stop in 2009 because it was so unpopular for Albertans. So people do say sometimes that like, oh, Alberta tried, it didn't work. Well, when they were trying, it was working, but unfortunately, it wasn't done in a way that made it accessible to people. Now what we find is that there are 85% of male mule deer, again, which are the most likely to be infected, right against that Saskatchewan border. So they're in a similar position to Saskatchewan, where there are very high levels of male deer who are infected. And they are reporting significant deer population declines as well, and their hunting harvests are decreasing, and hunters are unable to find older bucks when they're hunting, so also not great. Similarly, Wisconsin, so this is looking at how sort of that targeted kind of culling actually does work when we look at it from the empirical data. So it was first detected in 2002 in Wisconsin in the wild at a 0.51 prevalence rate. They undertook a sharpshooting program, really intensive. They went in really hard between 2003 and 2007. And you can see on that graph just there, we've got two different sort of colors. We've got the flat and the one that climbs up. So, and I'll talk you through it in a moment, that the little one that goes along the flat, that's Illinois, and we'll get there next. But the line showing there is when Wisconsin stopped doing their annual targeted cull after the hunting season. So previously they were sort of sitting around 0.94, just about 1% year on year. As soon as they stopped doing that program, it started to increase about 0.63% annually. So it just kept climbing. Some areas are now sitting at about 40% prevalence, which is not ideal. It's not as high as Saskatchewan or Alberta, but it is really significant, and they are also seeing population declines in those hot pockets. So this looks like a big flurry of blue dots on the map here. That is where it's considered endemic in Wisconsin. And I will talk about Illinois in a moment, but just talking about that next sort of piece on our arrow, management with hunters, but then some limited sharpshooting. This is Ohio and Indiana. Really briefly, I'm just going to run you through that. They have a limited culling approach where they only rely on mandated testing and hunter harvest. And then when they find a new detection of chronic wasting disease in a new area, they will do a targeted 1 mile radius around that animal in order to see how far it's spread and hopefully reduce its spread. So Ohio first detected CWD in captive deer in 2014, and then wild detection in 2020 via a taxidermist, with a really low prevalence level of 0.02. And at the moment, it does seem to be working. They're sort of targeted little radius of 1 mile, and they've only got a 0.56 prevalence. Remembering that none of these states are looking to eradicate, they're just looking to keep the prevalence low. Indiana is really new. They first detected chronic wasting disease in 2024, and they are also starting to do that 1 mile radius, but it's been a little bit too soon for them to be able to determine whether that is working at the moment because it's only been one season. Right, okay, back to Illinois. So we saw that blue map just before with Wisconsin. Below that, unfortunately, is red, that is Illinois. And they had an initial detection that had crept in from Wisconsin in 2003, it is considered, and that was 0.3% prevalence at that stage. Now, one of the things I want to highlight is that Illinois previously was considered to be the golden child of chronic wasting disease management. Look at them, they're amazing. They've kept it under such a low prevalence for so long, and it's true, they really did. Unfortunately, that has started to creep up in those really endemic areas. So, but I have to emphasize, they kept it under 2% for 16 years. That's really impressive, given that there are so many animals in a number of areas in that red on that map that are infected. So what did they do to do that? Well, they did an ongoing yearly sharpshooting program where they targeted areas of high CWD prevalence after the hunting season. They worked with hunters and they worked with landowners to get approval. They also considered it to be a mop-up approach, not replacing the value of hunters, but effectively filling up the rest of their sampling requirements by going out and doing it themselves. What they have found, though, is that it is increasing in the areas where it is endemic. So, for an example, between 2002 and 2010, that's 8 years, 80% of all CWD positives in the state were found from Boone or Winnebago counties. Those are the ones along the top where the red is. In 2020, that was only 5%. So obviously it started to get really strongly, starting that vortex, as you remember, started getting narrower somewhere else. So they have now decided to continue to be a poster child of response management, and they're changing their approach. They're being adaptable and cyclic and trying to figure out what is the next step, what do we need to do, rather than sticking rigid and being stubborn. How do we now change what we're doing to manage what we see rather than what we want to see? So they are in a new phase of their CWD response. They are now implementing a 5-year pilot because chronic wasting disease takes such a long time to see impacts of changes in management approach. They're doing it for 5 years, not just one. But they are looking at doing sharpshooting still in areas with less than 5% prevalence, so in new areas. And then within areas with more than 5% prevalence, they are doing hunter-controlled only, where they are really promoting hunter harvest by increasing permit allowances. I do also have to highlight that male prevalence, or prevalence in both sexes, is still under 10%, given that they've had CWD for more than 22 years. That's pretty impressive compared to some of the other examples. Okay, so Missouri is also another example. It's had it for a little bit less of time, but it's also a really good news story. So they had a detection of 0.11% prevalence in 2012. They have a statewide prevalence where you look at all of the areas that they're sampling of 0.4%, but that core area is about just over 1% prevalence. So that's still pretty good. That's 13 years of keeping it underneath 2%. They do regular post-season targeted removal of deer still within 1 to 2 miles of chronic wasting disease detections. They also heavily promote hunter harvest. Effectively, they give CWD management permits to landowners who have more than 5 acres, and it doesn't count towards their bag limit. So really promoting that ownership of land and encouraging those landowners to, one, hunt themselves, but also invite their friends to come and hunt. They also have a $75 processing reimbursement per deer, and they have what is called the CWD Hunter Harvest Initiative, which effectively means we want to get the hunters to compete with the government pretty much. If the hunters can meet the target, then the government doesn't need to do anything, which is awesome. So it's really kind of encouraging hunters to do all of the, not all of the legwork, but keep it in the hands of the hunters, not try and take it away or imply that hunters are not achieving what they need to achieve. So another good news story. Now, when we think about that arrow, we're at that red eradication end. So Norway did a pretty intensive eradication of reindeer. So this is Norway. They found CWD at about 1.2% prevalence in that top orange bit, so that's Nordfella, which I'm probably mispronouncing, in reindeer. And they decided, this is our one opportunity, it's new here. They did a lot of genetics, and they found that it was not related to any of the North American strains, which implied that it didn't come from being imported, it had spontaneously occurred in Norway. That's a bit of a worry. So they figured, you know what, this is our opportunity. If this is unique here and we've only found it in a couple of reindeer, let's just get rid of all the reindeer. We're going to get rid of it before it pops up anywhere else. So they killed more than 2,000 reindeer in one go, an entire herd. It took a really long time. There was a lot of public opposition, as I'm sure you can imagine. Went really well. They'd planned to fallow that area for 5 years. Then it popped up somewhere else. Ugh. It's not ideal. So they then found it in, I'm not even going to try and pronounce this, the area to the bottom, the larger, the larger orange piece, and that was the area that they actually wanted to try and repopulate the Nordfalla area with, but now those deer also have CWD. Really interestingly, they tried to make a perimeter fence around that fallowed area to try and prevent other animals going into it while they were fallowing it. Turns out reindeer also like to jump, so that didn't work either. So there's been a lot of attempt at being sort of the world first in Norway, and unfortunately it hasn't worked. One of the things to also highlight quickly is that there is something called spontaneous chronic wasting disease that has been detected in Norway, where it's only present in the brain, it's not present in the lymph nodes or the lymph systems, which interestingly means it cannot spread. They don't excrete prions. That is not something we found here, and it could be related to the fact that this is a completely different strain of chronic wasting disease. So interesting, there's a lot of biology that we still are learning about and still trying to figure out how we can use that to our advantage. Okay, I think I'm already really over time, so I'm just gonna go through this. So what can we take away from these examples? So we know for a fact that reduction in deer density reduces CWD prevalence. Hunter harvest is our biggest tool because again, it works as both reducing the number of animals, but also as surveillance. So therefore, hunters must be encouraged to continue to hunt in chronic wasting disease positive areas. That means we need quick turnarounds of testing. We can't have people waiting for months to know whether they can eat their animals. And the thinning of high density groups, such as urban deer, is really important before that prion load in the environment gets too high. We know for a fact that winter targeted reduction programs, sharpshooting, works to suppress CWD, but only if it's consistent. You can't just do it 1 or 2 years and then stop. You've got to be consistent with it. This is obviously easier in non-forested environments, and we need to target that around positive cases. We need to reduce artificial congregation, which we know about, and that it's pretty much impossible to eradicate, because not only does it spread in ways that we don't know about yet, it can also spontaneously pop up, as we know in Norway, and also what happens in Creutzfeldt-Jakob disease in humans, it can just spontaneously happen. We also know that widespread culling does not guarantee reintroduction. It's also really unpopular, so maybe we don't need to be so extreme. So looking at that arrow again, we probably want to sit somewhere between the management plus and the management plus plus, so management plus and a half maybe, where we have hunting and limited sharpshooting, whereas decided that it needed, or regular sharpshooting. And this is just based on the literature that we've seen. And so in order to get ahead of the spread, we need to reduce density consistently, and we also need to keep applying pressure. So, thinking that I'm over time, I was going to run you through how the harvesting works, but I think I'll save that. Effectively, harvesting works in a number of different ways through that non-selective or selective approach by getting rid of a number of different animals that help reduce spread in a number of different ways, with the overall argument being we need to keep doing it. Okay. Cool. So why do anything? So we know already that chronic wasting disease will kill deer prematurely as quickly as up to 2 years after being inoculated. We know that year-to-year survivability is halved or thirded in males. Sustained low prevalence does mean minimal population impacts, as we've seen in Illinois and some of the other examples. However, high prevalence will, and we've seen it in examples, we've seen it next door and we've seen it in Saskatchewan, that it will lead to population declines. Chronic wasting disease is a long-term disease. Impacts are not noticeable immediately, and I know that people in general tend to be kind of impatient. We want to know that the thing that we've done has worked immediately. The thing is, we won't with chronic wasting disease. So I really want to drive home that you might not see an impact of chronic wasting disease, but your children will, and your grandchildren definitely will. So if you let it go, it's irreversible. And so we need to make sure that reducing chronic wasting disease continues. So we need to reduce density now to maintain hunting opportunities in the future. And it sounds backwards, but you want to be able to provide the lifestyle that you guys love to your children and your grandchildren. More importantly, we want to keep our options open in case we get a vaccine or some kind of treatment that comes out. And just along here, I've got an example of the initial apparent prevalence for a number of different states, and just goes to show that just because BC has a really low prevalence right now, so did a lot of these other places, and now look where they are. So just having a low prevalence now doesn't mean that it's guaranteed for any particular length of time. I was going to go you through the impacts. I think you know. Effectively, higher chronic wasting disease means lower cervid populations. That has ecological impacts. It also reduces hunting opportunities, reduces funding where wildlife programs have been promoted or funded by hunting licenses, and it also reduces food security and food sovereignty for lower income or Indigenous peoples. So lots of issues. Really quickly, remember that vortex I was talking about? BC is really early in that vortex. Things are going really slowly at the moment, getting really low levels of CWD in a number of, huge number of samples coming in. Really early detection. There's no farmed cervids that we need to worry about in BC, which is awesome. Acted really quickly to implement regulations to reduce transmission, so around feeding and transport. Good surveillance numbers, and doing sessions like this with interested people and getting the word out about why doing something is important. So BC is in a really good place. I got in touch with Margot Pybus. I don't know if you know her. She's one of the technical disease experts in Alberta, and she said, and I'll quote, "BC has the best chance of any jurisdiction since the incursion has limited temporal, numerical, and geographic presence. Also, your province took a collaborative approach from the beginning, and this will help maintain social support, particularly important now that decisions are getting a little bit more hairy." And she said that she really wished that they went harder for longer in Alberta and they wouldn't be in this position that they are now. So final slide. Is it too late? No. Now is the time. Inaction now is irreversible, so if you don't do something now and chronic wasting disease prevalence starts to increase, it's really difficult to bring it down because you can never get rid of the prions in the environment. That means that you can't sort of sit around and say, oh, we'll just not go and hunt this year because that disease, we can wait. Unfortunately, you can't. This is the time. So maintain hunting opportunities now and in the future by acting now. We want to keep our options open, and so we want to, if possible, sustain hunting during the regular hunting season and during special hunts. This is my plea to you as hunters, which I assume most of you in the room are. Submit your samples, please, because if the government doesn't know where CWD is, they cannot adjust quickly, right? It ends up being like turning a tanker. It's too late. When additional licenses are released, make the most of them. Follow movement restrictions. Please don't take carcasses where you haven't found them or against the regulations. They are there for a reason. And be open to sharpshooting endeavours or targeted removals, because they're not necessarily there to replace you. They're there to encourage what you're doing to work in the longer term. So it should be in addition to hunting. So that's my very overtime chat for you. It took a really long time, apologies. So a big thank you again to our sponsors, really appreciate it. And I will open the floor to questions. So sorry about that. So long, sorry.
Thanks, Jess, that's awesome. Just before we open it to questions, just keep in mind Jess is not a provincial employee. She's not going to be able to answer questions on the provincial response. Holger will be talking about that, and you can ask those questions to him. And like Glenn said, just be respectful and courteous. And but yeah, any specific questions for Jess, we'll take now. Over here.
Great presentation. Could you tell me if you know anything about what they did in Wainwright, Alberta? I understand they allowed hunters to go. They would give them a tag to go get harvest a deer, and there was a CWD trailer there, and then when they brought the deer back, they were issued another tag to go out and get another deer, and they kept going. And my understanding, about 2 individuals harvested 100 deer in one go.
Wow, okay, so for those that didn't hear, that was a question around Wainwright in Alberta. And they had a process with a CWD replacement license. So if you caught an animal during a special hunt, you could bring it in, put it on a trailer, get a new tag, go out and catch another one. And there was approximately 2 individuals you mentioned that caught and hunted more than 100 deer in one session. I don't know too much about that. I know that I don't know that specific example. The replacement license was used in those early days as part of the increased hunting interest, and I believe that worked, but there was a lot of outcry from other hunters who found that it was unfair, that there were people who were taking advantage of that system. That being said, it was really effective from my understanding in ensuring that there were lots of deer coming in. They have now got rid of that, but I think maybe that's something that could be considered, but I would probably need to look more specifically at how that exact method worked. Thank you for the question.
We have one over here.
Yeah.
Yeah, my concern is these prions. You say when the infected animal defecates into the ground, the plant grows up through that, so another animal eats that, and then it gets infected, right? Is that how I understand?
Yeah, essentially, yes, that is correct.
So, last meeting I was here, there was a fella here that was a rancher. He goes and buys his hay from Alberta, which I know, I saw the map, there's hundreds of cases. So, the odds are when he's bringing that hay over here, it just so happens that he was hobby farm down south, and he's bringing that hay. How do they know? He said that how do I test again and see if I can have a positive case or not, right?
In the hay?
And they said you can't. So if we know there's a possibility that you can bring all this infected prions over here, why are we not stopping and saying, hey, we prefer you not getting your hay from those infected areas, or whatever vegetation that, or when you're buying a horse over there, he's eating over there, he comes over, he craps over here, these prions now are in the ground, the plants grow up, our deer are eating them, so we're not hitting it the full impact that we should. Yeah, we can call all the animals we want, but we need to stop it from where it starts and where they keep bringing it in. I mean, as far as BC, all I can see is the way it's slowed down is because of the Rocky Mountains. That's been a barrier as far as I'm concerned. But there is no barrier in the south of the border, and that's, of course, totally infected too. So the odds of some animals moving up here are pretty good, right? Pretty good.
Yeah.
So if the government is really serious, then they need to take it the whole way. You know, you need to say, hey, quit and bringing that infected stuff over here.
Yeah. So I think everyone probably heard, but it's a main question around what about fomites? So fomites are something that you might get a prion on something and then that gets moved over. So I know that the government at the moment has transport restrictions for carcasses and parts of animals, but it's a really good point. And if we want to be wholesome in the response, there needs to be restrictions on infected material that could be touched by deer in BC. So I can't comment, unfortunately, on whether that is being considered by the provincial government, but one of the difficulties to date is that we can't accurately test the hay at the moment, and this is a problem, right? There's a hole in our toolkit, is that we have that RT Quick system that's being developed in universities that's super sensitive. So as that gets more accredited and more able to develop, we might be able to take swabs of things like hay and then test it through there. And that way we'll be able to know who is considered a safe hay provider in Alberta versus those that might not be. So I think it's a really good point. I don't know where that's sitting in the provincial lens, but I completely agree. If we want to be like 100% about this, we actually have to be 100% about it. We can't let things like hay detriment what we're trying to achieve. Yeah. Oh, so many questions.
Just on the topic of going all the way, you did mention, you did in your talk, you did not mention New York State, which I thought had been the gold standard, that like Norway, used the nuclear option in 3 counties and eliminated every one of them. And the prion, there's only about 1% of the deer that were infected, but they eliminated them in a wide area. So and we managed to this was New York State, and it wasn't stopped by political pressure. They succeeded.
Yeah, I'm actually really happy that you've brought that up, because I purposely didn't include that because the case in New York State is quite unique. So what they found was infected wild deer right outside of captive facilities. So they were able to determine that that was a really quick, they caught it really early, and they effectively, as you mentioned, nuked a large area around those captive facilities. And it did happen, I think, twice, and it was successful twice. But they knew exactly where the reservoir of CWD was, and they got rid of anything that could potentially get it in that area, and then they put up fences. From what I've heard, I don't believe it has happened again. I don't believe that any deer has become reinfected from that, so it was a success. But at this point with chronic wasting disease in BC, we know that it's not from infected captive deer. So it is a kind of different scenario, but it is something to be aware of, that when it's available, we probably should be jumping into eradication if we can. Yeah. This woman.
Two quick questions. First is there a scientific theory as to why bucks are more likely to be infected? Second, is there any way to model, if there were no mitigation practices put in place, what a population would look like in 100 years?
Yeah, okay, so 2 great questions. So why are bucks more infected than does? This is to a degree affected by the species. So bucks in some species, particularly mule deer, tend to congregate together in bachelor groups where they interact with each other considerably more than doe equivalent groups. So they believe that this has something to do with it. They also tend to touch a number of other animals more frequently through ritual processes, but also with mating, and so they tend to be more accessible to CWD. So they have more interactions with other animals where they can be infected. That does vary from animal to animal. For example, in reindeer, there's also other things where male reindeer will chew on the vascular antlers of other male deer. Those vascular antlers have blood flow in them, and they will chew them down and effectively cause bleeding. So that blood, if it's got CWD, will then infect potentially other males as well. So more behavioural impacts. And then the following question was, what can we do about modelling to see what happens in 100 years per se? So this is all really hard to do with any level of accuracy. Some people are really interested in those resistant alleles, so those are resistant genes that allow some different animals to be more resistant to catching CWD than others. So looking at a number of generations on that super long scale level, between 100, 200 years, they think that the overall number of deer with those resistant genes will start to increase in proportion. That being said, it sounds great. Oh, if we just leave it, those deer are going to be resistant. It also means that you're going to have a massive genetic bottleneck because not very many of them are actually resistant. Remember, it's only a resistance, it's not an immunity. And so you do end up with, in all of those models, the proportion of those resistant alleles grows, but the overall population kind of does a so it does decline in almost all circumstances that I've read, but there is a difference in allele distribution in that population.
Okay, thanks everyone. Here to introduce Holger Bohm. Holger is with the BC government. He's a biologist stationed here in Cranbrook. Holger's been involved in wildlife management in Europe and Canada for over 20 years. He received a Bachelor of Science in Wildlife Management from the Van Hal Institute in the Netherlands, and his bachelor's thesis is on grouping behavior of European red deer. He completed his Master of Science in Ecology from the University of Alberta. Thesis there was Adverse Conditioning of Elk in Banff National Park. Holger's work has always been focused on large mammals. He's been, like I said, in the government of BC here, living in Cranbrook since 2015. First as the section head of the Habitat and Wildlife section in the Kootenay region, and now he's been the provincial ungulate specialist for the past 3 years. His focus is on aiming to use science to support management decisions, and since CWD has hit the area, that's been Holger's main focus over the past couple of years. So welcome, Holger, please.
Thank you. Is that good back there? No, I see, no, yes. Maybe, test. We good like that? There we go. I'm just looking at the one guy who's giving me the evil eye, so. Okay, hi. Thanks everybody, and thanks to Jessica for setting this up with all the background and what we've seen elsewhere. So I'm going to dive straight in and talk about what we've done in BC so far, what you all have been part of, and throw a few numbers at you, and get into a bit of what's the plan and what are we doing. So it's important to understand that we're still in the early stages of CWD in BC. As Jessica said, the disease has been around for quite a while, but it's only been here for a year. You'll see some of the numbers I'll present a little later show definitely how we are winding things up. We're getting new information daily. There's new test results coming in. There's new research. We talked about some detection methods and all that. So as we get things, we try to pass them on as fast as we can. This doesn't always go smooth, but we're doing our very best to pass on information because we're really aware, and I think Jessica made that very clear as well, that we need to be united in this response. Like, everybody needs to be on board. So it is really important that we work together on this. This map, I think, is very familiar to most of you. Those are the chronic wasting disease detections that we have to date. We have the 4 white-tailed deer and the 1 mule deer. Interestingly, the white-tails all around Cranbrook in that area much closer, and the mule deer further in the southeast. So I'm just going to go ahead and spoil a later slide. As of a couple of days ago, we have 1 confirmed positive in Cranbrook as well. So I'll show that on the slide and talk about that. I just know the questions have come up when people walked in already, so that's out there. So there's one more. So this slide is already outdated. So Jessica talked about prevalence a lot, and we talk about prevalence a lot. So I just wanted to take a minute to try and explain exactly what that means and how it can be deceiving. And Jessica touched on that a little bit, that depending on how you exactly report it, the numbers may change dramatically. So for our numbers, if we look at a provincial prevalence, so all the tests that we've done from voluntary and mandatory heads and all that, we have about 5,700 samples. We have those 5 positives plus the 1. And that gives us a prevalence of only 0.08%. No big deal, right? But if you focus a little further into the regional, the Kootenay boundary region, we have about 5,000 samples. The 5 positives brings us to 0.1. If we look at the management zone that we're really interested in, WMU 443, in there we have about 1,200 samples, the 4 positives out of the 1 year. So we are at 0.33. And then with Cranbrook, that was a little fun exercise, because up to this morning, I wasn't sure, we don't, and I'll get into this, I didn't have the results quite yet. Up to this morning, I only had 20 samples that I had the definitive results for, and we had the 1 positive. If I would have reported that out today, I would have told you it's 5%, right? 1 out of 20. This morning around 10 o'clock, we actually saw and received the other samples. So we have 94 of the 100 deer that we took out of Cranbrook. And again, I'm going ahead a little bit. And we still have that 1 positive. So more about a 1% prevalence. So just when we talk about that and when you see prevalence elsewhere, just ask a little bit, what does that mean? How are they reporting? It's really important that you don't just take the numbers for face value, that you kind of know where they come from. That is crucially important, and I'm showing a couple of slides here that Jessica just showed. When we look at what happened elsewhere, and this is what we're doing, we had Jessica on the team at the beginning to talk about exactly what she did talk about here. The pattern is very clear in all jurisdictions. The prevalence at the beginning is a flat line. So I did the little red circle there for Alberta between 98 and 2008, somewhere in the point something, maybe 1%, somewhere there. Same in, if you look at Illinois and Wisconsin, so there's a flat line at the beginning. And the good news is that we are in that flat line right now. So people have been asking, oh, there's only 5 positives. What's the big deal? The big deal is that we are in that flat line. As Jessica, I think, had made sufficiently clear, every single other place has gone up eventually. So we have an opportunity here to kind of get there and work on this as hard as we can right now. Another question we get a lot is how many samples do you need? Where do you go? Where do you get the samples? And this graph is a little messy. Unfortunately, I got a shot only of the full graph. It's from Dr. Michelle Bohm, I guess, epidemiologist. And it explains how many samples you need in a population of what size to determine the prevalence of what. So I'll walk you through that. Don't be scared. At the bottom, you have the minimum chronic wasting disease prevalence to detect. So if we want to detect at 0.5, 1.0, 2.0, or 5.0 prevalence. So let's say we want to know if we are at 1. So that would be the second line that divides all those graphs. Then at the top of each graph, you have the population that we're looking at. So the smallest in the top left would be 100. The biggest would be 5,000 for this exercise. And then you have different colored plots, which is the test sensitivity. Our test is pretty good. So our sensitivity is at about 99%. So we're looking for the purple dot. So you can see that in a population of, let's pick as a little exercise, 500, for a 1% rate, we need about 220 samples. So that's half the population. For a population of 5,000 at 1%, we also need about 250, so much less of the population. So these numbers don't really jive with me anyways. Maybe someone has a more mathematical brain than I do. But it's people, like especially when we went into Cranbrook, people go like, oh, how many do you need? Why do you need that many? And the smaller the population gets, the larger the percentage of the population you need to test to find that animal that is sick. So the extreme example is in the top left, a population of 100. You can do the math yourself. If you want to detect a 1% prevalence, it's 1 in 100, you got to kill them all, pretty much. So small populations require large sample sizes. So in summary for that, on the prevalence, the early detection that we had, it's an opportunity. We can use that information that we're getting for informed decision making. We need to communicate very clear. We're working on that. We're always trying to be better. We're not great. And important to know is that animals don't believe in lines on a map. If we calculate the prevalence in 4-3, and there's like a case just next doors, the animals will cross over. So we got to be very careful with what we calculate where and what that means for the neighboring MUs or regions or provinces. In the end, knowledge is power, and again, I think Jessica made that very clear. The more samples we have, the more confidence we can have in the prevalence. You saw that in that graph with all the dots. And we definitely need that support to get our hands on all these samples. And this is where hunters come in, and I'll be harping on this all evening long. We need those samples, so bringing those heads, it's a team effort. We all got to chip in and get that so we can make those decisions based on good data. There's a chronic wasting disease management plan, which Kate Nelson, who most of you I think have seen before, she has been working on this much longer than I have. And she had a plan ready before we found the first positive, which is awesome. And I'm not going to go through the whole plan. If you want to put yourself to sleep, you can read it in the evening. It's very lengthy, but it's very detailed. The initial response objectives in there are the key items. And this kind of jives with the arrow that Jessica showed in her slide, the what do you do when. And the important part is to determine the chronic wasting disease status and do we have positive cases? Check. What species do we have affected? We know we have the 4 whitetails, the mule deer. Check. And then we are looking at those local areas. Where did these positives happen? And what are we going to do in this area? So I'll talk about the special hunt in a minute. And then the goal is to reduce the disease transmission and the spread. So step 1, and you've all been part of this, is the mandatory head collection that we put in through the regulations, which I like to apologize for. Regulations are always difficult to make, and they're always difficult to word right. And whenever we think we got it nailed, someone calls it up and goes, what in the hell does this mean? And then we go like, oh, we got to make this better. But we were able to get regulations out and put the mandatory head collections in there. And it's been working really, really good. We got a lot of heads in, and which gives us great confidence in those samples. So this is the numbers of the mandatory head collections just in the chronic wasting disease response area in both years. So since day 1, pretty much at this point, from the mandatory head collections, we have 4,682 samples, and we have those 5 positives. Plus the Cranbrook one that didn't make the list yet. Which is really great, like to have that many samples. So you can all give yourselves a high five. This is amazing to get that many samples in. I'll tease this apart in a moment, a little bit more. Just to give the rest of BC a heads up or like a thumbs up as well, there is voluntary head collections pretty much anywhere else. Not that this is set up well everywhere. It's not like here we have the freezers and we are set up for these collections. We're working on that. We're getting better. We're trying to get more freezers. But we also have the opportunity, like you can pretty much drop off a head in almost any office anywhere, and which gives us a chance to detect those randoms that show up. And Jessica talked about a few of those where just randomly, like something popped up. So this is a chance for us to catch that. We're like really trying to focus on this area, but it's good to have those voluntary heads there as well. Another source of information is roadkill. So the province hired a roadkill coordinator. So there's a phone number out there, and she's working mostly with First Nations, Ministry of Transportation, conservation officers, trappers, municipalities. We've been really trying to get that phone number out there. It's a bit of a balancing act. We can't really have everybody who sees a dead deer call her. She'll do nothing else because 15 people drive by the same dead deer. So the pathway, through Ministry of Transportation is a little more smooth like that. Working out the kinks, but that's again over this fiscal year alone, about 188 samples just from that. So those are animals that died anyways, and we had a chance to, because we were somewhat organized, to pick them up and get those tested. So just in comparison, and again, this is like kudos to you. In the first year, so fiscal year, we work by fiscal years because we government. In 23-24, we had 802 heads sampled and submitted. We had that first positive in that fiscal year. And then we brought in the mandatory head collections and we talked to everybody, and you guys did awesome. And then the next fiscal year, the numbers look like that. So remember those numbers in a minute, 802 versus 3,400 when I talk about testing times. But most importantly, and I think again, Jessica gonna put a finger on that, I also assume most of you are hunters. Hunting is the most important tool for chronic wasting disease management. We need those samples in. We need to know where the disease sits, how many animals are diseased. So every management plan that you read, hunting is mentioned in there. It's a tool in there. The samples come from the hunters. And they not only bring in the samples, but they also reduce the density of those animals on the landscape a little bit. So using hunting as a tool, I think most of you are aware of the special hunt that was put in place around those positives around Cranbrook. It's like a little bit, so Cranbrook is that gray blob in the middle, and then if unfortunately I couldn't pull together a map with the stars and the outline on there. But the green outline is the special hunt area that we put in here just about a month ago or so, which responds to the positives around Cranbrook. So if you remember, Jessica talked about in some jurisdictions that did a 1 mile radius. We have decided on like a 5 kilometer radius. And it so happened that the positives were aligned around Cranbrook, that if you connect those 5 kilometer radius areas and you take out Cranbrook, that you get this area. And then the weird shape is based on what can actually be enforced. So this is working with the conservation officers, because if you just draw a line on the map, nobody knows where that is. So this is adjusted to like the height of land, similar to your regular hunting regulations. So 118 permits were issued for that special hunt. Any species, any sex. We had 36 heads submitted that were good for testing. There was a few extras that didn't quite work out for different reasons. I don't need to go into that. Nobody's fault, just sometimes it doesn't work out. But 36 were submitted, which means that the success rate of the hunters that went out was about 33%. So what we're working on right now is, or maybe I should go to the bottom first. At this point, we have 18 negative results back and 18 are still pending. So as I said, the results are coming in daily. We also wait a little bit sometimes, like everybody, and that's where we're at right now. But the key part is that part of the permit, so I'm looking at everyone who had one, was to give us a call or report if you were not successful, and why not? Did you not go out? Did you not see anything? Because that gives us a great idea of how many animals are there. Why did only out of 118 permits. Why did we only get 36 heads? Be great if we can get that information, because with the number only, we don't know if there is only 36 animals there and you got them all, or did you pass on some because they didn't look cool enough, or what happened, or they looked sick, or what's going on. So that's that communication piece both ways. Let us know what happened out there so that we can use the information and work with that. I just wanted to throw that in here. So there was also, we had the regulation change that you're aware of. There was also the general order, which is done by the Ministry of Agriculture and Food. That one is addressing the movement of carcasses, and I know there was a lot of struggle with that. We had lots of phone calls. What does that mean? Like, I cut the spine out, leave that here. Can I take the meat? Which part? We know there's room for improvement. The aim of that is, and I think the hay question and what Jessica said is like a great example of that. The idea is to find that middle ground between not moving things to places they shouldn't be, but also make it somewhat feasible. Like, we can't tell you to carry a whole elk out of the bush. It's just not going to happen unless you're like Arnold or something. And right, so it needs to be somewhat feasible. So we're trying to kind of find that line. So hoping to work with you, stakeholder groups, First Nations, everybody who's involved, see if we can improve on those first cuts. And this goes again with, we're in this together, we're trying to work on this together, and we have to develop this together. So let's kind of figure this out. So hopefully we can make this better. Moving to urban deer, the slide everybody was waiting for. So there was a press release, I think, a little bit before that, saying like the province is going to go out and remove and test urban deer, February 18th to 28th, which we did. And the team in Cranbrook was quite successful. They really honed in on this population and got the 100 animals that we targeted. As of today, we have 93 negative results, 1 positive, an adult female, and 6 that are still pending. So that doesn't mean they're positive or anything, it just means they didn't get tested yet. They're somewhere in the queue still. So waiting for that. The good news on this graph, on this map, is that we were able to achieve quite a spread. So we got samples from just about anywhere in town, which was quite a feat. So kudos to the team that went out and did that. Lots of long nights, really hard work. There was a lot of people involved removing those animals. There was a lot of people involved getting the samples out. So really happy it worked like that. Of course, a little unhappy that we got that 1 positive, but in all honesty, if you look at the other 3 positives in that area, that placement is not really super surprising. We also targeted Kimberley, same time frame, so it was a really, really tight window. Kimberley is a little further removed from where we have the positives at this point, of course, but it is the next high density population. And again, referring back to Jessica's talk, we really are worried about those high density populations, because similar to you going to a party and there's someone has a cold, everybody's going to be sick next weekend. Same for deer. They are in town and they all rub noses and they all stick together. If CWD is there, it's going to burn through that fairly quickly. And we are worried about the cities as a source population. So if you have all these animals there, they migrate out of town. The bucks come in and out of town. I'll talk about that a little bit more in a minute as well. They may carry that disease into the wild and then just create new vectors, like to spread the disease from town. Unfortunately, we had some run-ins with some activists. We only got 26 samples. I'll spare you the details. But at this point, we have 15 negative and 11 pending. Remember the graph with all the dots? This is not enough samples for us to actually say something about Kimberley. We still don't know, not enough samples really. So hopefully we can find a solution there, and so we can actually do some proper sampling in Kimberley. A word to testing. Again, I know there was frustrations, and we're working on that. Just wanted to run through what happens when you drop a head in. So you drop the head in the freezer, then the trusty team collects the heads from the freezers and brings them to the facility in Cranbrook. And then the tissues get sampled. So there's people like Kate here actually painstakingly taking the head, cutting it open, finding the sample, and then they get sent to the agricultural lab in Abbotsford. So there's like a bunch of bottlenecks there already. So every single head needs to be tested, like pulled out manually. All the samples need to be put in little packages. And then they can only be sent to the lab once a week because they cannot deal with samples like all the time. They need to get them fresh, like once they're defrosted. So really there's only 1 day a week that gives them enough time to receive the samples and then do something with them right away. So there's a few timing issues there, which when you drop off your head on a Friday and shipping day was Thursday, there's like a week right there that your sample is just gonna sit there. So once the samples are there, at the beginning there was literally one guy in the lab testing all these heads. So there's that ELISA test that Jessica talked about. And then if that comes back non-negative, and it's called non-negative, then that test gets sent on to the federal CFIA lab and to confirm that result. So there's a bunch of steps. So if you remember back to how great you were at getting the 3,000 some samples this year, up from 800, there was a lot of heads coming in all of a sudden. So that's why, and I do apologize, why we saw delays that we really don't want. So we're addressing that. We have expanded the sampling capacity. We've brought in more people. We are currently working very, very hard on getting a better, bigger, and nicer processing facility in Cranbrook. The folks in Abbotsford have upped their staff, and we've also reached out to other labs in Saskatchewan and Washington. So if we get like an onslaught, possibly, hopefully, next hunting season, that we can kind of distribute the samples to other places as well to get those results back. We are absolutely aware that the test turnarounds are really what are going to make or break the bank here, because we can't have you wait for your sample for like 6, 8, 10 weeks. Which is a rare case. I'm not going to promise you a week or two. We can all dream, but I'll just tell you we'll try to do better. We're trying everything to get the next hunting season samples to you as fast as we possibly can. Well, thank you. I'll use that opportunity to have a drink. So let's talk a bit about research. So there's a guy, Adam Ford, most of you probably have heard of him, know who that is, at UBC. He's a professor there. One of his students, Jacob Hubner, I think, has also, before chronic wasting disease already, has been very interested in animal movements. Where do they move? How do they move? And this has become super crucial for us to know those movement corridors, because that'll be important to know how the disease will most likely spread. So on this slide, you can see all the information, quote unquote, that he has used to create a movement map that I'll show you in a minute. So these are all the red dots are mule deer locations from radio collars. The blue dots, whitetail deer from radio collars. And then the little crosses are the positive chronic wasting disease cases. And he stuffed all that into the magic machine and created a circuitscape map, which is what you see here. Which looks pretty crazy, just like that. But if you just imagine that The more yellow you see, the easier it is for a deer to move through that area. So the darker, the more intense the yellow, the more movement there is. So I'll do what I'm not supposed to do and walk towards the screen. But you can kind of see nicely how the valleys kind of come out, like through here. But you can also see some east-west connections. So those are the areas we think and we plan for. We think about how we believe that disease is going to probably spread its way through BC. Important to know here that cities were excluded from this map because you can kind of see in those little inserts, they just come up as like a black dot because nobody knows what the heck is going on in the city. So this is focusing on the animal in the more natural areas, and it depicts their movement there. So you can kind of see from Cranbrook where this is going to go. Very interesting to us right now is that area just south of Creston, because there's those 2 positives down there. And that's going to be, like if you're a betting man and you want to put money on something, that's probably where we're going to find the next positive sooner than later, unfortunately. So once again, I'll go back to the hay question. Can we stop it at the border? No. Animals don't believe in lines on the ground. It's really just not happening. We can do our very best. We can just be ready for it. And so we've been planning on what happens if this pops up in Creston. It's quite convenient, honestly, that we have the office here. It's a little more difficult in Creston. So that's something that's going through our minds right now. I did say that the cities were excluded. So let's talk about cities. We removed those deer, as I said. We are fully aware that cities may act as a source for the disease once the disease is there. But we don't have a map like I just showed you, that circuscape map. We don't really know how animals move in and out of cities. Nobody really ever cared. We know anecdotally, and I hope you can confirm that we see more bucks in town in fall. And they seem to be coming in and moving out, which we are really concerned about because that is exactly the risk that we're facing. Like if they come in for mating season and then they bugger off, take the disease with them, that's how a city may work as a vector for that disease to get into the environment further. So the regional team here around Emily O'Donovan went out and found a little bit of money, and they collared 10 bucks in Cranbrook to see how they move in Cranbrook, with the hope of doing 10 more in fall, and we're also working on getting some callers out into Kimberley. Here's a map she provided. Unfortunately, the black lines don't come out very well, but you can see the white dotted lines are the home ranges. So this is all the animal dots. If you use the radio collars we put on, take a location like roughly once a day, and if you just plot all of them, and you can kind of create a cloud, and then it shows you where that animal moves. So that's what we call a home range. So each of those little circle, oval, whatever the shapes are, is the home range of one of those animals. And the black lines that you can probably not see all that well are the mule deer, and the white lines are the white-tailed deer. And you can kind of tell that there's much more overlap than we thought there is. Especially down in the south, you can kind of see how the black lines and the white lines overlap quite a bit. And you can also tell that there's quite a range in home range sizes. Love our little buddy on the golf course there. He's just living there, so not really going anywhere. So there's this has just started in January. So the data I just showed you, there's really only 3 months of data. Not a lot of movements yet, so those home ranges will change, maybe dramatically in fall. We'll see. 11 callers in total, and quite interesting too that when these animals were caught, 8 of them were in poor and 3 in what we would call emaciated, so really skinny condition. So I'm not throwing that in here because they're bucks and it's the end of winter. They're supposed to be skinny. We get a lot of calls of people who mean really well. Oh, there's this deer and it's really, really skinny. It's like, yeah, it's March, they're skinny. They don't have chronic wasting disease necessarily. But it was quite shocking to see the team reports that how in what bad condition these animals are at this time of the year, in town even. I mean, you'd think they live in town because there's a lot of food, but they're not doing so hot. So more to come here, more callers to go out. And again, that current collaring window, only January to March. We'll get more data, we'll report back on that. Other research, and again, Jessica touched on that. Why do we have to kill these animals to test them? It's not great. I'm the first one to agree with that. Are there non-lethal and non-invasive testing methods? Yes. So Jessica already talked about the RT-Quick, which is like a very, very highly sensitive tool. It's not something that we have the labs for, if I understand correctly, or the pathway for to actually use those results yet. Hopefully that's going to come soon. There's other teams that work on blood tests. There's fecal tests that work. You can do biopsies. It's always a little questionable if you have an animal in hand already to take a biopsy. Do you let it go again because it may have the disease? So you have to catch it twice if it was indeed sick. And then a lot of these methods are not super accurate, and Jessica talked about those false negatives that we really don't want. We need a method that is very, very good, that really tells us if it is a true positive or a true negative. So RT-Quick seems to be the method that we have the most hope in right now. We're not quite there yet. Yeah, so to date, the only reliable test is unfortunately still the collection of tissues from dead animals. Put the BSE cow in here, just as a reminder that this is not something that is only in deer and in cervids. There's other teams working on this. So like, especially with BSE, there was human casualties. So there's like a lot of labs trying to figure this out as far as how can we test for this? How can we get better at that? So lots of research done. We need to look a little bit beyond of what's done on the deer populations only. As far as the human health risk goes, I think Jessica spoke to that as well. There's no evidence at this point that the disease can be transmitted to humans. However, and I just want to highlight that again, she said, I think you don't want to be the first one. I agree. I don't want to be the first one either. So to prevent the potential risk, wait those 2 or 3 weeks until the test results are there. 4, if you have to, just don't risk it. It's not worth it. Just don't eat that meat until you have that negative result. Just as a reminder, and I think we've talked about this before as well, cooking it very hot or having your deer steak, not medium rare, but well done, is not going to do the trick. Like, you need thousands of degrees. Like, you really don't want to eat that meat anymore when it's charcoal like that. So the path forward, there's no beating around the bush, and I like Jessica's little bit of red. This will spread. This is not going to go away. We got to live with it. We got to work on it. We have an opportunity because we are, remember the graphs that I showed, we are in that flat line. We are at the 0 to 1% prevalence somewhere, depending on which kind of math you use. So we really have an opportunity here to stay on top of that. We are aiming to manage to the lowest possible prevalence. At this point, we are, if you remember that arrow that Jessica showed, we are in the middle somewhere. So we're doing hunting, we're having the special hunt. We are considering bringing in sharpshooters if the special hunts don't work. Not there yet, but talking about it. We need to continue and learn from people like Jessica and summarize what's been done in other jurisdictions and see what worked and what didn't work. continue monitoring, continue bringing your heads in, continue hunting in those areas, even though you may have to wait a couple of weeks until you can eat your meat. And we have to be flexible. So adjust those plans, adjust the regulations, come with better wording for the general order, do all that. So hoping to have that communication work with you and with First Nations, get that input, see if we can kind of work this out together so that we can guarantee that long-term buy-in from everybody. Because I think, again, Jessica has shown as soon as we lose that, we're screwed. So we got to work on this together. And with that, there's all the teams, all the teamwork and all that. Lots and lots of people that are involved. And I'm open for question if Jessica left me enough time.
Obvious question everybody's going to have is why, if we're the only area in the province that's been found to have the disease, why do we not have a testing facility here to speed up the process?
That's a very good question. Like building a testing facility is not something you can do in a month or two. So one of the thoughts was bringing a facility here. There is a lot of, from what I understand, I'm not someone who does like asset management, a lot of red tape as far as permits and what like the fume hoods and what is all involved. It needs to be specially sealed and all that. So at this point, it is easier to up the Abbotsford capacity and just deal with it that it's there and lose that day or two of shipping and kind of work with that than building something here. But it's most definitely something that we have considered and we will consider again if that's necessary. Yeah, good question. Like in the end, it would probably save us the shipping if we get the labs set up properly. Definitely on the list, but it's not something we can build overnight. Yeah.
Are they going to come and get the rest of the ones in the cities? And secondly, including in the walled communities, the strata places. They're hiding in there.
I hear you're a fan. So yes and no. So given that we have that positive from Cranbrook, unfortunately, I was really hoping that we have all results. It's a little bit of a, so there's no release out yet for that one test result because we wanted to wait for everything so we don't have to do like, oh, we've tested 50, here's the results. We tested another 20. So we're going to do like a release of all the results once we have them, hopefully in like a week or so. So based on that, we will have to, again, adjust and decide what we want to do. Quite honestly, unlikely we're going to do something immediately, just because it's fawning season, and that's morally and ethically difficult. I don't want to speak too frankly, but I think if or when we do something, it's probably going to be fall, early next winter. As far as the disease management goes, definitely interested on continuing to work on those urban deer because they are a problem for disease spread, absolutely. In the back there?
Yeah, are there any plans to expand the sampling to Sparwood, Alford, Invermere, get ahead of the game?
So we kind of monitoring the samples that come in. Oh, sorry, the question was, is there any plans to expand the sampling to Sparwood and other areas? So again, looking at what comes in for samples, like the voluntary ones, and kind of keeping an eye on things. We're definitely open to expanding that at any time. We need the resources, both financially and in human power, to put all the freezers out, have sampling teams, because you can't really make people drive all that way. So there's a lot of logistics behind that. So we want to be carefully, surgically with like where we do that, and so we don't run everybody into the ground. But yeah, absolutely. If in a perfect world, we'd have testing everywhere. But we just need to see how much we can do at what time, where. But it's definitely on the list to consider. Yeah.
Yeah, Jessica spoke to target density and where they were able to mitigate it. So So is it the same for all species? That, like deer, whitetail, and elk? And what is our current density?
That is a very, very good question. The question was, what is our current density and what is the target density? And unfortunately, as opposed to elk and moose, we are fairly decent at counting out of a helicopter. We're not doing so hard on deer. They're elusive little creatures, and they're really difficult to count. So traditionally, our estimates for density on deer populations come from hunted samples. So there's a bit of math involved. It's like, well, if, you know, X number of hunters, which is why I'm going back to that, it's important for us to know, did you go out and did you get nothing? X number of hunters went out, X number of deer came in, X number of deer showed up at butchers. There's like a bit of an algorithm. We're like, well, we're probably in that realm. So we are fully understanding we need to find ways of determining better how many deer there are. It's very difficult. So I can't tell you what the density is. We have like a rough idea just from captures and collaring projects and those remote camera projects, but nothing good enough for us to really, for me to tell you a number right now. So we are, yeah, we're trying to look into exactly that, like what is the target density, what is the number. So it seems like, again, the gold approach, the suggested best approach right now is to really focus on those positives as opposed to trying like a broader density reduction. So going in, creating those special hunts, working with the hunters, and then considering like if the hunters are not successful for whatever reason, do we need to bring in a team to take out a few more animals out of those hot zones? I think that's probably the path where we're going right now. Yeah, John.
Holger, tied to that question about populations, if we take a higher hunt, that means there's a lower population going into winter and a lower population. So next year we've got fewer deer, and then the next year we still need samples, whatever the percentage is. I'm just wondering what the math does in terms of, it seems to me that the math is going to necessarily lead to significantly reduced deer populations in a relatively short time, especially if you're trying to get enough samples in order for it to be mathematically useful. The less animals you have, the more samples you need, and so on. So I'm just wondering how that all fits together in terms of the long-term population curves.
Yeah, that's a very good question. So again, things in progress, things we're trying to figure out. At this point, we have an extra hunt only in those areas around the positives, right? So if you remember that little green zone, the special hunt, so that was created with that 5 kilometer zone in mind around each positive to really go in and have like an added effort in there. At this point, we haven't really gone and opened the Kootenays up, hunt anywhere, everywhere, whenever you want. That's not in the plans right now. So there's no general population reduction. We're really focusing more on that approach that was, again, in that middle of Jessica's arrow there, trying to get into those positives and reducing and pulling out those individuals that we know or are much more likely to be diseased. So with that, hoping to keep that prevalence as low as possible. Whatever number that is, we kind of trying to figure that out. It seems like 1%, 2%, 3% has worked elsewhere. Maybe we can beat, what was the record? 16 years, I think they held it. Yeah, so hoping we can do something like that. Yeah, over here. I don't know, you guys, your buddies, go for it.
Are you aware of the 2 deer that were found dead in, it's on 6th Street, pardon me, yes, 6th Street and 16th Avenue. There was a property owner there, when the snow melted, there wound up being 2 deer on his property. So I'm not sure if your office was aware of those 2 deer. And then one of the other gentlemen that comes to the pool was telling me that if you want more deer, he counted roughly about 18 deer congregating at Laurie Middle School
Yeah.
At 9 o'clock at night.
Yeah.
So there's an easy spot.
Yeah. There was a specific question about a couple of deer. We have that roadkill coordinator, so she works with the cities. So if that gentleman that has those 2 dead deer on their property, hopefully they would have called the city.
They've already been taken or something.
Perfect. So if they went to the city, they would have come to us. I can't track every particular deer. And yeah, and the other question had to do with there's someone who reports 18 deer in a certain area, and yeah, like even though we removed 100, there's still plenty of deer in Cranbrook. Don't you worry. Yeah.
Yeah, maybe your answer to this might simply be no comment, but the
If you're sat like that,
the 100 when you got 100 deer called in Cranbrook somewhat restores my faith in Cranbrook, but given that you're down to 26 in Kimberley, the question that haunts me is were you encountering organized obstructionists there, or were these individual actors?
Well, let's go with the no comment on that. It felt rather organized. Let's leave it at that. And everything else you have to find on Facebook or wherever else people put things. It most definitely got to the point of where we called the police. So
yeah. Doesn't that put really a wrench in your picture of what you're trying to do with the
yes,
samples?
it does. Like that's why we can tell you, again, if you think about the samples we need to show a certain prevalence, 25, not enough. So we don't really know what's going on in Kimberley. Yeah.
We have one more question, and then Jessica said she will stay for those that have specific questions, and I think Holger.
Oh yeah, I'll be here all night.
One more question in the public, and then we'll.
All right. Would you be able to provide that number for the city so more people can call that? I can tell you an experience I had. I had a dead deer on my property. I phoned the CO service. They said they wouldn't come and get it.
Yes.
So they told us to phone the city. We phoned the city, and they said, we're not coming to get it if it's on your property. If it was on a street, they would do it. And they said, if you want to drag it to the street, maybe we'll come and get it. So then I phoned the CO back, told him what had transpired. He said, oh, well, I don't know what to tell you. You're going to have to figure it out.
Yeah.
So then we phoned the city back, and they said, well, even if we come and get it, we just take it and dump it in the bush. So that the information that was given to me by the city was that they weren't following the rules that were.
Please tell me that was more than like a couple of months ago.
No. It was a month ago.
All right, so
I phoned the CO back and explained it to him. He said, oh, I guess we better tell the biologist what the cities do.
Yeah, I don't know how to, I'm sure everybody heard the question. There's a bit of problems with how we share information between the municipalities and the Conservation Officer Service. So that roadkill coordinator is trying to patch all that up.
Can you get that number out to the public?
Yes and no. So the problem, I kind of alluded to that, is if I give that number out to everybody, like she's going to get, again, 50 calls from everybody who drives by that one dead deer, like between Kimberley and Cranbrook, right? So the process right now is, you can test that, don't test that, but you can call the city now. They should say, yep, well, we know who to call. So if that worked, then we golden. So that, and we've seen this. It's one of those things we have to train people. So we've seen this work better and better. So hopefully we'll get that. And I've myself gone out with her like a bunch of times, and we go onto people's properties because people can or don't want to drag a dead deer onto the road. That looks kind of weird.
They told us that they weren't allowed on our property, so the city wouldn't do it.
The city, what?
Yep.
Okay, next question.
We'll make this the last question.
Sure.
The prions are in the spine and the brain, and they're in the blood, from what I understand, as well. So what about the predators, wolves and bears and that? Are they prone to getting this too? Because if they're eating the carcasses, they're leaving their feces out, they're transporting it too. Is there any plans on, in your scope of things, looking after them too, to eliminate or reduce the spread of it?
I know exactly what you're after. I'm going to answer that, and you chime in if I'm spewing. Yeah. No, so the question was about predators when they eat like a diseased carcass. What happens then? So first of all, it's specific, and stop me anytime. It's specific to cervids, so predators can get the disease, but they can most definitely work as vectors. So if they eat something and then they poop somewhere else, and then they can spread it in the environment. So definitely a concern. And again, I hate bringing up the hay thing again, but it's like one of those things like, yes, we're also thinking about that. Right now, we have definitely other problems on our hands, so we need to figure that out. And then in the long term, we can kind of see what can be done there. There's, I think at this point, it's safe to say that it's not like very high on the list of concern. Talking to the roadkill coordinator, she goes to the highway pits where all the animals are collected. There's, like every time she goes there, there's hundreds of ravens. So you do the math, what happens there, right? So this is where I talked on one of my slides about the feasibility versus like, we need to find, we can't start shooting all the ravens, right? Because one raven may pick up a piece of meat somewhere. There's somewhere we need to be in a realm of where we can actually do something and not go insane.
Thanks, Holger.